Good morning and welcome to today's webinar, Effective Control of Anthracnose in Turfgrass, presented by PBI-Gordon and taught by Brandon Horvath, Ph.D. My name is Lisa Wick. I'm a Senior Manager of E-Learning Programs at GCSAA, and I will be today's moderator. A couple of quick housekeeping notes before I turn it over to Brandon. We are recording today's event, and access to that recording will be sent as part of the follow-up email. Your audio is muted in this system. We encourage you to ask questions as we go along. You can type those into the question and answer box in the GoToWebinar control panel. If your control panel has minimized, you're going to see a narrow strip with an orange rectangle at the top. Click on that and that will open the control panel and you'll be able to type into the question and answer box. You'll also see there the handout that's available for you to download. Some of you heard me mention this looks a little bit different than our normal PDF, as my remote computer wasn't real happy with making that today, so I apologize. Today's session is eligible for GCSA education points, and at the end of the program, we'll give you the code to use on the association's website. We hope you find the webinar valuable today. It's brought to us in partnership with PBI-Gordon. With a full line of herbicides, insecticides, fungicides, growth regulators, and other products, PBI-Gordon Corporation is a national leader in the professional turf and ornamental management industry. Brandon Horvath, Ph.D., is an associate professor of turfgrass pathology in the Plant Sciences Department at the University of Tennessee and is the 2020 Tom Samples Professional of the Year as awarded by the Tennessee Turfgrass Association. Horvath received his bachelor's degree from The Ohio State University. He received his master's and Ph.D. from Michigan State University. He has worked with championship golf courses, conducts research in the area of turfgrass disease epidemiology, and has taught over 250 presentations and published numerous articles about his work. Please join me in welcoming today's presenter, Brandon Horvath, Ph.D. Brandon?
Thanks, Lisa, and thanks to PBI-Gordon for sponsoring the webinar. Hopefully we'll have some time here to kind of explore some thoughts about anthracnose control, and we're going to spend some time talking about the disease and its disease cycle. As Lisa mentioned, epidemiology is an interest of mine, so obviously with the times that we're currently living in and everybody becoming an epidemiologist apparently, I'm not going to make any reference to the coronavirus because that's not the kind of epidemiology I'm familiar with, but rather turfgrass diseases and how these diseases develop and expand are kind of the area that I spent a good deal of my time studying, and anthracnose was the subject of my master's thesis looking at, and I'll refer to that throughout the presentation as well. In the chat box or in the questions box, I kind of have those open and we'll be kind of keeping tabs on those. If you have questions, make sure you feel free to ask. It's good to see some familiar names on the attendees list. I kind of scrolled through it here a few minutes ago and saw a few familiar names. So nice to see a few of you that I recognize, and those of you that I don't know me. As Lisa said, I'm at the University of Tennessee, and we'll go ahead and get started here. So let's see. So there's my Twitter handle at UTTurfPath. Feel free to follow me on Twitter and ask questions. If you have questions that come up after reviewing this or watching this and you want to come back and ask a question, feel free to reach out to me on Twitter at UTTurfPath, and then at UTTurfgrass is our turfgrass program Twitter handle. And if you have questions about our overall program or you want to see what some of my colleagues like Dr. Brosnan or Dr. Sorokin are doing, feel free to follow us on at UTTurfgrass. POA Annua Live, those of you that know Dr. Brosnan know that he hosts a seminar, and it's applicable for this anthracnose seminar because POA Annua is one of the big hosts for anthracnose, and so a lot of you may be interested in tuning into POA Annua Live. As a result of some of the things that we've experienced here, that's been canceled. So there won't be a Facebook or a POA annual live presentation on the plots from Dr. Brosnan, but the POA Annua Herbicide Resistance Project that's sponsored by SCRI are going to host a workshop at that same time on March 31st at 10 a.m. And so make sure you pay attention to @ResistPOA on Twitter for further details on that. So that's going to kind of swap out and they're going to do a workshop on herbicide resistance, particularly with regards to POA annua. So if you're interested in that, that will be another educational opportunity while we're all kind of sequestered at home for the most part. Keep that in mind if you're interested. And as I said, I'm in Knoxville. Lisa, can you remind me, can they see my pointer?
Yes, sir, we sure can.
Okay, great. So I'm right here in Knoxville, and so one of the cool things about being in Knoxville is that that green band that you see going across the country there is what constitutes the bulk of the transition zone, which is of course the area kind of between where we have the cool climate from the warm climate and the kind of the demarcation zone between which we grow cool season grasses and warm season grasses. And being right smack in the center of that, kind of in the central part of the US, means that we have the ability and the opportunity to grow just about every turf grass that you can imagine. We have at our research farm, we have a 1/4 acre of St. Augustine grass, which is typically grown way down south. We keep it alive over the winter with some covers. It is super susceptible to everything as a result. And I've been dumb enough to grow perennial ryegrass, which I like to call the white mouse of the turf world. So we can grow all those grasses, and as AJ Powell liked to say, and one of the things that we teach our students is that we grow all the grasses equally poorly. And what that really means is that at any time during the year, whatever grass that we're managing is going to have a period of time where it faces a weak period where it's unable to fight off the challenges to it. And so then that's where all the diseases occur. That's where all the problems kind of develop. And so as a pathologist, it's a great place to do work because we don't have to try really hard to get really spectacular epidemics of diseases going on just about any grass that we want to work on. And so that's a nice thing in terms of looking at things like anthracnose and other kinds of problems, because it's relatively easy for us to get it year in and year out and see what works. When I give a presentation in person, I usually ask the audience what this is, and we're not going to test the chat box here to do this, but
I know it's a triangle.
That's it. It's just a triangle. It's just a triangle, and it's important. Like one of the reasons why I hammer on this concept is because in almost every textbook and almost every place that you see this, you see the labels of the different parts of the disease triangle at the vertices of the triangle. So you see host, pathogen, and environment, and they're placed at the vertices. And the problem with that is that the original author of the disease triangle paper that kind of postulated this idea that this combination of these three things results in disease had the labels on the sides of the triangle. So they had like environment, host, and pathogen, or wherever you want to put your labels. And what he really talked about was that you can kind of conceptualize the area of this triangle as being the potential for disease development. So the bigger the triangle, the more potential there is for disease development. And if we do something to alter the size of that triangle, then we have effectively reduced or increased the potential for disease development. And so, for example, if we chose a resistant host to plant on a new establishment, then we're going to shrink, say, the side of the host part of the triangle, and that makes the triangle smaller. And what that does is that every practice that you engage in, regardless of, and we're going to talk about a lot of things that have nothing to do with fungicide today, every practice that you engage in, from cultivation to top dressing to mowing to air movement, etc., is something that you want to make sure that you're doing so that you can reduce the size of that triangle and get that triangle as small as possible. So the when we think about fungicide control, fungicides control, roughly all things being equal, about 90, we can get 95% control out of most fungicides if they're applied properly at the proper timing and that kind of thing. So if we think about that 5% that might be left over, say this small area here, kind of the tip of one of these bits of the triangle, then if we had 5% left over on this triangle, that's going to be a smaller amount than 5% left over on this triangle. And what that constitutes is kind of what we would see as breakthrough, if you will, right? Like if you make an application of something and you see it give you pretty good control, but you still see something break through, that's a hint that's telling you that maybe culturally or mechanically there's something that you haven't quite accounted for in that overall potential for disease development, and that's why you're seeing some breakthrough. If you can get that triangle small enough, then the breakthrough, if you will, that 5% that's left over, ideally will be small enough that you don't ever really see it. And so that's an advantage, right? And so if we shrink the environment as well as the host, then we get a smaller triangle still. So that the whole concept of this is something that I really want you to kind of ingrain in your head, is thinking about like every practice that you do to manage the surfaces that you provide is effectively changing the size of this triangle. And if we add time into it, we're going to talk about time being an important factor here, then that also has a role to play in terms of disease development, because that you can kind of think of that prism being extruded from a triangular, like a press that's like Play-Doh press, right? You're pressing out the little spaghetti strings, and instead of a spaghetti string, it's a triangular string. And instead of that triangle being the same size all the time, it's constantly changing with all the practices that you're doing. And what we're trying to do is we're trying to cut off the development of that disease over time, because there is a time component that plays a role in how these diseases develop. And for example, with anthracnose, that time component is the time that it takes for this disease cycle that is very important for disease development to occur. And so a spore has to land on the leaf surface, and you can see this is an anthracnose spore here. It's landed on the leaf surface, it's germinated, so it produces this short little germ tube. And then you can kind of see this dark kind of oval shaped pad, and that pad serves a couple of functions. The first thing is it has some sticky kind of sugary type of material that is exuded out of it that allows it to stick and stabilize itself to the leaf itself, so that it's not going to get dislodged if water or something comes along and tries to wash it off. And so it's going to stick to that leaf surface. And then the reason why it's dark is that it's melanized. It produces melanin, very much like the melanin that we produce when we expose ourselves to the sun. And that melanin hardens the surface of the hyphae and allows it to generate a tremendous amount of pressure. And then you can kind of notice, especially in this image here, that aprosoria, that little lobe there, is right on the edge of two cells. And the reason for that is that it will push a tube out of the bottom of it and kind of spread those cells apart without breaking them and slide into the intercellular space in the plant. And that allows it to get into an environment that is very nice and very comfortable for its development. And so this is where the disease cycle for anthracnose starts. And this is all happening at a microscopic scale that we can't directly observe. We haven't seen symptoms yet. All we're seeing is, all we have are these spores landing on leaves and germinating. But that is a time point where if you interrupt the disease cycle here, disease never develops and you never see symptom development. And that's really what we're going after, is trying to make sure that when we do have a management intervention of one kind or another, we're trying to interrupt this time course of this disease cycle. So let's see. There we go. So so this is what happens. So this is hyphae that is growing intercellularly, and you can kind of see, you know, that like there's a cell wall right here. You can kind of see, and the hyphae is growing right beside it and kind of growing up and around other cells in the plant. And you can kind of see it making a network around cells. And that's very much what happens with anthracnose. Anthracnose is in a group of fungi that we like to call hemibiotrophs. And what hemi means is half or partly. Biotroph means that it uses a living organism for its food. And the reason why it's a hemibiotroph is that it's also a little bit of a necrotroph. And a necrotroph means that it uses a dead organism. Necro is dead, and it uses a dead organism. So what happens is that germ tube germinates, produces the aprosorium, the hyphae comes in here, and it grows intercellularly, and it starts using some of the exudates from plant cells and nutrients from the plant cells to continue to fuel its growth. And it will take over and produce a network of hyphae in the plant. And again, this is before you ever see symptom development. So this is all happening before you see something that says, oh, this might be a plant that's not doing so well. And then once the plant starts to undergo stress, and this is one of the reasons why this has always been kind of a controversial disease, is that, you know, some people say it's a bona fide disease and that it, you know, you can control it with fungicides, and so it's not stress related. And then there's other people that would argue that it's very stress related and that you would never really have this disease if you never go under stress. And you guys all know that in reality, there never is one of those two extremes, right? There's never an extreme where you have zero stress. Well, maybe, you know, if you don't have any golfers out there and you're closed right now, you might be about as close as you can get to the best possible outcome, but you're always putting the plant under some sort of stress. Mowing stress, drought stress, moisture stress, traffic stress. You go down the list of stresses that the plant can experience, and it's responding to those stresses by changing its physiology. And so when that happens, when this fungus has infected an area of the plant, then that fungus will sense via the production of some of these compounds that the plant produces when it's under stress, that the plant is under stress. And that's when it begins its necrotrophic phase, and it starts producing enzymes and other things to start degrading cell walls and start taking over cells and sucking up all the nutrients it can, because it realizes, hey, this plant isn't going to be around that long. I need to get everything I can get so that I can reproduce. And so then this is what we start to see. So this is, we have kind of two phases of disease development that we're going to talk about. The first phase, or the first disease that we think about, is what we call foliar anthracnose. And this is a disease primarily of the foliage. If you look at a bentgrass plant, you can find it on the surface. This is one of the diseases that we often see on our bentgrass plots in Knoxville, both on putting surfaces as well as you see it a lot on fairways. One of the main additional stressors that I've seen where we see problems with anthracnose on fairways is quality of cut. Oftentimes when I'm looking at a sample of anthracnose under a microscope, I'll see a lot of frayed edges of the leaves. And so everything you can do to make sure that that quality of cut is as high as it can possibly be helps reduce the potential for this disease. And this is the symptom that you see. So you can see there's a drainage tile here, a drainage basin, and you see these areas of kind of brown tissue that just kind of look like something's a little bit off and we're not exactly sure. And when we go in and we look at it, we start to see some of the hallmark signs of anthracnose. This is from my turf plots. So you can see there's turf that looks nice and green, and then you see this kind of orange cast. And when you zoom in on that orange cast, that's where you start to see structures that clearly indicate that this isn't drought stress or something else. This is kind of bona fide anthracnose. And this is another good example of those plots. And as that disease progresses, we start to get areas like this where the turf starts to thin, and you start to see clear symptoms, but they tend to still be a diffuse kind of pattern. They're not generally not in any kind of straight lines or circular patches. They kind of they're kind of a diffuse pattern, and the reason for that has to do with how this disease moves, and it primarily moves via water splash, and we'll talk about that in a minute. So if we get closer, we've got this area that's thinning. We get closer still and we zoom in, then this is what we tend to see in that foliar anthracnose situation. So we see all of these little black, pepper grain type structures on the leaf, especially on browning leaf tissue or tissue that started to lose color. And then you can also often see it on green leaf tissue, but it's at a much earlier stage of development than this. And these black structures, I always think that they look like, if you've ever been swimming in a tropical area and done snorkeling or anything, you see sea urchins all over the place. That's what I always relate it to, is that it looks like sea urchins on the sea floor, is that you see all these little pin cushions of little black structures with needles sticking out of them. And if we zoom in a little further, let's see. Oh, there we go. So this is why I think of sea urchin, because I think it looks very much like a sea urchin. So this is a singular acervulus, and this is the spore producing structure of anthracnose. And this spore here is not the one we're paying attention to. These big circular spores are a different organism, but all these little, look like little fingernail or little half moon kinds of shapes, these are all the spores of anthracnose. And you can see just off of this one acervulus, there's hundreds and hundreds and even thousands of spores that are produced by a singular acervulus. And like I said, these spores are rain splash dispersed. And you can see this is a microscope slide with a drop of water, and that drop of water has dispersed these spores off the surface of the acervulus, and now these are out and able to move and find new places to infect, right? And if we look at it a little bit differently, you can kind of see this pinkish brown mass here. Those are all the spores, and then these little black hairs of the acervulus are what we call setae, and those spores we call conidia. And when they're in the acervulus, they're kind of enmeshed in this like jelly-like matrix that has compounds in it that prevent germination. And so again, once you start kind of working your way through it, you realize that this makes a lot of sense. So a raindrop comes, it splashes, or an irrigation drop comes and splashes some spores off of this acervulus. That washes the spore clean of the germination inhibitor, because you don't want the spore germinating when it's in the fruiting body, right? That makes sense. So it washes it clean of that germination inhibitor and splashes it somewhere else. And if the conditions are right, it will germinate, produce its aprosorium, and start infecting. And so that's kind of how the cycle continues. So then if we get it, particularly on annual bluegrass, on Poa annua, but we can see it on bentgrass as well, if this disease is insidious enough and gets going enough, then oftentimes what we see is we don't see the foliar phase of the anthracnose. What we end up seeing is actually a phase of the disease where it infects what we call the crown, or the growing point of the plant. And this is much more damaging to the turf surface, because that crown, that growing point, is the point at which that plant can rejuvenate itself. And oftentimes, even with the worst foliar diseases that we have, if given enough time, that plant will recover because you haven't killed the growing point. So it'll reproduce leaf tissue and it'll regrow out, it just might not look so good for a little while. Well, with crown rotting anthracnose, it's a different story. That part of the plant that rejuvenates the plant ends up infected, and then that plant dies. And as the disease progresses, we start to see these same kind of copper color, but we just see these areas that look a little bit off. And as they develop and spread, they get worse and worse. And if we were to go in and look at them a little closer, we can start to see some acervuli being produced up on the foliage. But if we get closer still, and I'm going to scan through this slide here. So there's the acervulus on some anthracnose on annual bluegrass, and you can see that pink. And then if we pull those brown plants out, what we find is the crowns are black and rotted, and this is very classic kinds of disease from anthracnose. And these images are from an article that I'm going to reference here in just a few minutes. This is just very classic crown rotting anthracnose. And the reason why this is insidious is because this is where the growing point, the rejuvenation point of that plant is, and it's all infected with the fungus, and that tissue is dying. And that's part of the reason why these plants will pull very easily out of the turf. And if you catch it early, you have an opportunity to kind of get ahead of it. If it gets bad enough, then it becomes very difficult to control, because all of those spores that are being produced here are being disseminated by irrigation water and rainwater to other places, and you're getting more infection. And so it takes a while. I always, kind of talk about, disease development in anthracnose is a lot like taking a golf lesson. You usually get worse before you get better. And that's very typical of what we see. So anthracnose is caused by the fungus Calototricum cereale. We used to call it Calototricum graminicola. That name was changed fairly recently. One of the things that we like to do in science is switch names up for you. So Calototricum cereale is a separate species from the anthracnose species that infect corn and sorghum. And there's some question and some evidence that suggests that the anthracnose that infects bentgrass isn't necessarily the same anthracnose, same species of anthracnose that often infects annual bluegrass in the crown rotting kind of phase. And so that's still open for debate. But we talked about the stress. Stress is a big part of the disease cycle here. Stress, when we think of that, we're talking about mowing stress, moisture stress, drought stress, temperature stress, traffic stress. All of these things are going to present challenges for you in terms of managing anthracnose development. It's one of the reasons why I think this got quite out of hand as we moved into the 2000s with lowered mowing heights and increased expectations. And we can certainly see this on annual bluegrass as well as creeping bentgrass. And so all of those stresses are all a part of this disease development process, and it's an important cycle on both of these grasses. Annual bluegrass is far more susceptible than creeping bentgrass. We tend to see crown rotting anthracnose on creeping bentgrass, particularly on older bentgrass greens that, in a lot of cases are no longer around because they've been switched to Bermuda grass, and particularly a little farther south into the southern part of the transition zone where bentgrass is kind of under stress throughout the summer. We don't tend to see it as much on northern bentgrass that's grown farther north. We tend to see it much more prevalently on annual bluegrass in that situation. So, as I said, the article that those pictures came from, and what I want to point you to is a golf course management article that is really in my opinion, one of the best examples of a continuous effort to really tease apart what's going on with regards to this disease. And those of you that may be from the Northeast certainly remember this this period of time in the early 2000s where anthracnose was a major issue on annual bluegrass in the Northeast particularly, but all over the country. And this article that came out in 2012 in GCM kind of showcases years of concentrated effort by Bruce Clark and several others, looking at, and it continues to this day in a lot of cases, looking at what are the best management practices that you can employ to reduce that overall potential for disease development. You're not going to get rid of it by just employing some of these practices, but you're going to shrink the size of that triangle down to where your fungicide management options become far more effective in terms of what you're trying to do than if you're not engaged in any of these practices and you're relying on what comes out of the nozzle to try to give you that control.
I want to interject here for just a second, Brandon.
Sure.
That GCM article, normally I would have as a handout given this situation. I don't at this point, but we will have that for follow-up.
Okay, great.
Also, Bruce Clark's work, as you said, has been ongoing. He's been looking at anthracnose for years. In fact, the first webinar that was done by GCSA in 2003 was him and Jim Murphy on anthracnose. He has updated that with different research that they have found over the years. And so if you're interested, he does have an on-demand that's done about the same time that this particular GCM article came out, that people can access.
Yeah, that's great. And Alex, it looks like you have a question. You got your hand up. Feel free to type in the chat box what your question is, and I'll try to answer it in line as we go. So feel free to type that question in the chat box.
He's working on audio. We're trying to fix that. Sorry.
Okay. That's the question. Okay. Very good. Okay. So let's see. So there we go. So all these best management practices serve as kind of guideline to kind of help you think about if you are struggling with anthracnose control. These are things that you want to look at in your program to see, like, how can I make things better? Okay, so one of the things that this work showed very clearly across, not just at Bruce's place, but at multiple locations that examined this, that spoon feeding and nitrogen fertility is much better overall in terms of disease development than monthly applications. And if you think about this just kind of logically, it makes lots of sense, right? You have a plant that you're managing at a fairly short height of cut. That plant has what we would argue is a somewhat diminished root system in the sense that it certainly doesn't produce roots like bentgrass. And so it's a shorter root system. So anything you can do to kind of give it the fertility and the food that it needs to grow on a regular basis, rather than kind of a feast or famine kind of approach, is going to reduce that overall stress development and not go through periods of too much nitrogen, then not enough, then too much, then not enough. It's that cycling back and forth that helps kind of tick off this stress kind of situation that leads to overall disease development. So spoon feeding with 1/10 of a pound of fertility weekly is much better than the 4/10 of a pound monthly. 2/10 of a pound weekly was better than 1/10 of a pound. And some of you might go, oh, well, what about green speed? And, you know, over the last decade and a half or two, there's been lots of work done on nitrogen fertility and green speed. And what almost every single one of those papers has shown is that you lose about 4 inches of green speed for every pound of nitrogen you increase. So when we're talking about going from 0.1 to 0.2 pounds, what we can expect is a couple of inches max of green speed that we're losing. And so we want to be very careful about thinking about using nitrogen for green speed management. We want to use our mowing and our other practices to try to manage green speed, rolling, to try to manage green speed and use fertility to maintain the health of the plant and provide it with enough nitrogen so that it can go through its physiologically required reactions using nitrogen as a part of those molecules that get produced in order to conduct growth and reproduction of its cells, so that we can maintain that growth even in the face of some disease attack. So 0.2 of a pound weekly, better than 1/10 of a pound. And then if you're in a situation where you have some disease that's active, some of the work that Bruce did showed that upwards of 0.4 to 1/2 a pound of nitrogen per week can help reduce those symptoms over the short term. So, and again, I've got those parentheses, not for summer use. You want to make sure that that's in that shoulder part of the season, in the spring or in the fall, where you can see some of this crop up. If you want to recover quickly, there's an opportunity for you to boost that fertility a little bit and really rapidly grow. And then in terms of nitrogen source, ammonium sulfate, not quite as good as urea, ammonium nitrate, calcium nitrate. So ammonium sulfate showing a little less disease than urea, ammonium nitrate, and calcium nitrate. So if you're using an ammonium sulfate source, you can see some reduced disease development. The challenge with that is always potential for black layer, first of all. So we got to be careful about that. And we can also see some challenges from some other diseases where a nitrate or a urea source might be a little better as well. So you want to make sure that you're paying attention to not just the nitrogen rate, but also the nitrogen source, and using nitrogen sources that make sense during different periods of the year for diseases that may be active during that time, because there are some slight subtle differences. And I wouldn't say that the nitrogen source choice is a primary consideration. It's really more of a consideration that if you've got everything else kind of dialed in and you're still seeing a little bit, then this is something you might look at in terms of, is there something there that I can change? Also, we've seen some evidence that if you're deficient in potassium, you can see some increase in severity. So making sure you're doing soil tests and properly following things like MLSN or other soil fertility measures to make sure that you're never deficient in those key nutrients is important for anthracnose management. So you definitely, the other thing that we've seen is that oftentimes we think of a fall kind of a dormant feed, and some of the work that Bruce did several years ago showed that particularly with anthracnose management, shifting that fall fertility to the spring in order to encourage the plant to grow readily when it's in a period. If we think about annual bluegrass, again, we're in that latter part of the winter coming into the spring. The plant wants to produce seed heads, which is of course the scourge of all of you that are managing POA. And it's using all of its resources to put that seed head up. And so making sure that it has plenty of fertility to continue growing, especially if you're managing seed heads, that it has that energy to continue growing is really important. And that often leads to a reduction in overall disease severity throughout the season. So that's something also to consider doing, is moving that fall fertility to a more spring type front-loaded approach. Granular applications, fine in the spring, fine in the fall. Quickly transition to those spoon feeding applications as you enter the growing season, so that you're continually replenishing that fertility for that plant so that it can maintain its growth. Top dressing is another area that when I worked on this when I was a master's student, there was a lot of thought that we were damaging the plant, and that damage to the plant was resulting in increased anthracnose. And so one of the things that I was trying to do in my master's project was I was trying to damage plants in the greenhouse repeatedly and reliably to get good infection, and often struggled with it. And not surprisingly, some of the work has borne out that top dressing itself is a beneficial practice to reducing the severity of this disease. It's really beneficial. So if you're making top dressing applications and you think you need to stop when you see disease, my advice to you would be to not stop, because that top dressing actually insulates those crowns and protects those crowns and growing points from the traffic stress, the mowing stress, and the rolling stress that you're putting onto those greens. It provides a cushion that helps keep those crowns intact and under less stress than if they were sitting right up on the soil surface and then you were rolling right across the crowns because you had stopped top dressing and putting them in that kind of upper matrix of soil. Aggressive programs, I've got that in quotes, 400 to 800 pounds of sand per 1,000 square feet every 14 days has improved turf quality. So there's been lots of research done by Jim Murphy and Bruce looking at that top dressing applications. Definitely want to work your sand into the canopy with a method that is not as injurious to turf. There's lots of different ways to do that. Irrigation is one, cocoa fiber mats rather than a stiff brush, those kinds of things. What I would encourage you to do is to find a technique and a strategy for you that works with your management practices that you've employed at your facility to be able to work that sand in a way that doesn't damage the turf. Because there is definitely some evidence to show that if you damage the turf, you're adding to that stress, and then that can, you know, kind of help pop off some infection. It's important to maintain summer top dressing by reducing rates but increasing frequency. So we're still putting out similar amounts of sand. We just want to make sure that we're putting that out on a regular basis, and that's what's led to the dusting type of applications. And then I would encourage you, I was up in Canada a couple of weeks ago before all of our travel got shut down, and got to hear Dr. Murphy talk about some of his current work on finer sands and top dressing, and that work comes directly out of the anthracnose work, and then some of the questions about working that finer sand into the canopy a little more easily than some of the coarser sands. And so Jim has continued that work, and I would encourage you to follow that very closely as well if anthracnose is something you struggle with, because it's there's a lot of really good information in his work on how you know, how fine is too fine, and that's a particularly important consideration in terms of upper surface soil moisture and firmness, which I know all of you are concerned about. And so his work is continuing to shed light on what are the best practices for top dressing, and I want you to make sure that you pay attention to that.
Hey, Brandon, before you go on, there are a couple of different questions that have come in, and a couple that relate specifically to what you just talked about.
Okay.
In low growing rates.
Where are they at? Because I'm not seeing them in the
well,
chat or
they're in the question and answer box, which you don't see. But let me read them out to you, and that way they get captured on the recording.
Okay.
In low growing rates, we should do top dressing, light top dressing in the same way.
So I'm going to assume that when we talk about low growing rate, we're talking about the plant is not growing readily.
Right.
And in that situation, what I would say is what you're trying to do is you're trying to match top dressing amount and top dressing frequency to the growth of the plant. So if you are growing very slowly, then that's going to mean that your top dressing amount is going to get lower, less volume or less amount of sand applied. And then I would say that you still want to maintain frequency. So that's where we often see, especially as we get into the summer, those very, very light dusting applications of sand. But you want to maintain that cushion of sand around the crown, regardless of how fast the plant is growing. So I suppose if you're in a tournament situation and you've got the plant growth down to almost zero, where it's just barely kind of replenishing itself, but it's not really growing beyond where it was, and you're just barely regrowing the amount of tissue that you cut off the day before, then that would be a period where you might want to extend the frequency of that sand application a little farther. But that's a pretty rare occasion compared to a regular kind of low growth period under normal maintenance considerations where you're still growing, you're just growing slowly. And so you want to continue to apply sand to maintain that cushion, albeit at a lower amount, but I would maintain the frequency. So that's a great question. What's the next one?
Our next one is any associated disease problems caused by additional N applications in spring.
Can you say that again?
Yes, any associated disease problems caused by additional N applications in spring.
That's another really good question. I know a lot of you often worry about, well, if I apply more nitrogen, I'm going to make the plant more susceptible to things like brown patch and pythium. And what I would say to that is that in the amounts that we're talking about, which are fairly low, and often many of you are well down at the bottom of the barrel in terms of total nitrogen for the year from what most academics would argue is kind of sufficient for plant growth, largely because of concerns about green speed and things like that. The increase in fertility, if you're struggling with anthracnose, is not going to be enough to then light off like brown patch or pythium. Most of the work that was done that we typically associate with, oh, increased nitrogen means more brown patch or increased nitrogen means more pythium. If you look at the rates in a lot of those papers that were done back when we kind of developed that type of thought process, it was back when we were following Dr. Beard's book in the mid 70s and 80s, and we were applying 10 pounds of nitrogen per 1,000 square feet per year to bentgrass and POA, and 12 or 13 pounds of nitrogen resulted in increased brown patch and pythium. So when we're down, at 1.5 pounds or 2 pounds for the year, applying 2.5 pounds or 2 3/4 pounds of nitrogen is not going to result in a dramatic increase in the amount of brown patch and pythium that we see. Great question. Any other questions that have popped up?
Iron sulfate insoluble, can that help decrease anthracnose pressure?
There's been some work to look at iron applications and particularly very high rates of iron applications. My experience and my advice with those kinds of things are that you want to be very cautious and you definitely want to do your homework. There's some anecdotal evidence that things might be decreased, but I'm not sure that anybody has looked aggressively at the impact that those iron applications have on anthracnose directly. And oftentimes with that darkening of the plant, you can sometimes mask something that is still occurring. You just don't see it because you've darkened plants up dramatically. And so with iron applications, that's not an area of expertise that I have. And so I would just say that you want to be very cautious and you want to talk to people that are fertility experts and then look for and see what evidence out there that you can find in the turfgrass information file on iron applications with anthracnose. But my familiarity with that literature is that it's fairly scant, mostly because there's not a lot of funding behind doing those kinds of pieces of research, looking at things like iron, because it's hard to get a proprietary product out of iron application. Everybody has an iron product, and so looking at some of those things, you're kind of doing the homework of some of your competitors as well. So I would just be very cautious with that, but there is some evidence, some anecdotal evidence that those kinds of applications can help reduce it. Anything else for now?
One last question that I missed earlier is, can this be seen through an eyeglass or do you need a microscope? And I'm guessing they're talking about things like what you've got on the screen there.
Yeah, like a hand lens. Yeah, you can definitely see things like this with a hand lens. You can see this with your naked eye, and then the acervuli, the little sea urchin things, with a hand lens or a macroscope or something in the field, you can put that right down on the turf canopy, and looking through it with 20 or 30 or 40x, you can definitely see those structures and see that that's occurring on the turf canopy. Great question, and I highly recommend something like a macroscope that gives you 40x. It's like a little mini microscope that has a clear shield you can put right down on the canopy, focus it, and you can see stuff like the acerbuli development on the surface of the plant. And then if you pull plants out and you see these blackened crowns, that's a pretty clear hallmark symptom that you've got anthracnose. Yeah, great question.
All right, continue on and we'll take more questions later. Thank you.
Okay, great. Let's see. Okay, so yeah, definitely follow those recommendations. So irrigation wise, one of the things that I think has changed in the last like 12 or 15 years and is largely responsible for some of the improvements in turf quality we've seen over the summer is the use of the TDR probe. And if you're not using a TDR probe to manage soil moisture at your facility, I would really encourage you to do that. So moisture management in anthracnose is critical, and really understanding how to use TDR probes is a really important thing. I have talked with a number of superintendents that believe that they're using the TDR probe to keep the turf just above that wilting point where you got to go out and hand water. That's certainly one strategy, but what I would encourage you to do is to make sure that moisture stress is never an issue. And when I say moisture stress is never an issue, what I mean is you never want it to be too dry, and you also never want it to be too wet. And so what that means is that you want to maintain soil moisture kind of in the middle of the road from field capacity, where the pore space of the soil is completely filled and you're freely draining, down to wilt point. And so one of the ways that you can do that with a TDR probe is on a wet, rainy day where everything's been raining, you got kind of a day that it's been raining consistently all day, go out to your drain outlet, notice that it's fully draining. You can see water running out of the drain outlet from the green. Take your TDR probe, go up and take a measurement. That's going to give you some number. If you're on a sand-based green, it's going to give you some number somewhere in the 30s or so. Depends on organic matter and how many soluble salts you have and all that kind of stuff. That's relative to your golf course. So don't pay attention to the number down the street. Don't pay attention to your buddy's number. It's the number at your facility that's important. So measure that. And then in a period of dry weather, let the surfaces dry down until you just start to see some wilt, and measure right near that wilt, right in that area of wilt, what the soil moisture is there. And you're going to get a number somewhere in the single digits or lower teens, and you're going to say, okay, that's say 11%. Okay, so if it was 33% for the high end and 11% for the low end, then we want to be somewhere in the middle, somewhere around 18 to 22%. That's kind of where we want to hang. And if we're in a tournament situation, we might want to be a little lower than that. If we're in a situation where we're trying to recover plants and we're growing readily, we might want to be at the upper end of that. But what we don't want to do is we never want to be so dry that the plant is constantly wilting, because that's a stress. And we never want to be so wet that there's no oxygen in the root zone, because that's a stress. So we want to be in the middle where the pore space is kind of 50-50 moisture and air. And generally speaking, we can use our hand watering and irrigation at 60 to 80% ET to really dial that in and make sure, as I say at the bottom, that water is never limiting the growth of your turf, because that's how you minimize the amount of moisture stress that you place on those plants to keep those plants growing effectively. Verticutting and cultivation, very much like top dressing, you don't need to be overly concerned, but you do, if you have active disease, want to try to minimize turf damage as much as possible. So that might be a period of time to stop a verticutting or a grooming practice, but under normal conditions, it's not something to be overly concerned. Okay, generally speaking, whenever we cause damage with these things, it's usually when somebody's rushing and they're not paying attention to the process. And so you want to make sure that you train your staff to do the things that they need to do to make sure that that operation is done properly. Mowing and rolling, as we talked about throughout the presentation, a big piece of this, particularly when it comes to stress. The work that Dr. Clark and others have done has shown pretty consistently that anything below 125 starts to really dramatically increase stress on that annual bluegrass and result in possible potential conditions for anthracnose development. So 125 is kind of a starting point in terms of if you have anthracnose and you're struggling and you're mowing down at 100 or 095, then you need to start thinking about raising the heights up a bit and using double cutting and rolling. Because what's interesting is, and this has been done over multiple years, multiple times by different graduate students of Dr. Clark's, is that increased frequency of mow and rolling frequencies have not increased disease severity. So you can use that 125 benchmark as kind of a base height. They've also shown that when you increase the heights under active disease to 0.14 or higher, you can raise that height a bit and use double cutting and rolling to maintain speed, and you end up with true surfaces that aren't suffering as a result of the disease development. But when you start getting below 125, that's really where disease starts to develop. And so that's a key kind of take home here, is that if you have, or if you're struggling with anthracnose on your surfaces, you definitely want to look at what you're doing with mowing and rolling, because that's been a key driver in terms of the amount of stress that those plants experience and how to best go about reducing that. Because again, remember, we're thinking about that triangle. These things aren't going to get rid of disease. If we don't spray a fungicide and we just raise our heights, we're going to get less disease, but we're not getting rid of it. But by taking that stress off and allowing the plant to continue to grow and photosynthesize, we're going to have a better chance that the fungicide is going to work better, and that's going to give us higher quality surfaces. And at the end of the day, I would argue that a surface that is true and is uniform and looks good is going to be much more important to your membership than a modeled surface that looks unwell and has thin areas and areas that are growing okay and might be a little bit bumpy as a result. All of those kinds of things are things that folks don't like. And so you can maintain quite a bit of speed with double cutting and rolling. And the research that they've done has shown that those kinds of practices, by increasing frequency, don't decrease or help maintain ball roll without increasing disease severity. So that's something that you want to pay attention to. And then we've got just a few more minutes. So PGRs, that was another one that would come up quite a bit when I was doing research on anthracnose, and still comes up from time to time. Generally speaking, PGRs don't increase disease, but some materials may help reduce disease, but you can't count on that. Okay, so like paclobutrazole, it's kind of a very light fungicide, and so sometimes that can help reduce disease. But at the end of the day, if you're on a PGR program, and I say here, if you're on Primo, stay on Primo, that's what you want to do. Everything you can do, and this last bullet point is critical. If you have to control seed heads as much as possible, so you need to dial in your timing on seed head control as best as you can. We're right in that window for a lot of you in the Northeast, and we're getting close to that window getting past here as we start to warm up. So you've got to make sure that you control those seed heads as much as possible, so that you go into the spring and summer growth period with a plant that hasn't used a whole bunch of carbohydrate reserves to push out a seed head. And so that is a key piece to reducing disease severity. Fungicide wise, so now we talked about all of those practices that are involved in cultural practices. Let's talk about fungicide control. So the cool thing is we've got lots of tools to manage this disease if we've done the right cultural practices. If we don't do the right cultural practices, then we're going to find ourselves in a pretty good pickle trying to manage disease with just the fungicides. But the DMI fungicides have been very effective on anthracnose control. Generally speaking, we want to make sure that we water most of these in very lightly. We want to get at least 1/10 of an inch of irrigation down. Some work from Dr. Kearns at NC State has shown that if we don't get 1/10 of an inch of irrigation down to water fungicides in, that we don't get it down into that upper root zone, which is really where we want this to be, so that it can be translocated towards the crown of the plant. And so we want to make sure that we get that 1/10 of an inch down. The DMIs, flutriafol, Rayora, a new fungicide, new DMI, Torque, and the other tebuconazoles, certainly effective. Tridimethon, mefentrifluconazole, which is Maxtima, new fungicide from BASF, all with good efficacy on anthracnose. The SDHI class, which has been a kind of a hot area for us in the last couple of years, penthiopyrad, which is Ballista, fluxapyroxad, which is Exemplar, fluopyram, which is Exteris, Tekin, which is isofetamide and tebuconazole, all excellent for anthracnose control. And then the QOIs, pyraclostrobin, azoxystrobin, fluoxystrobin, Insignia, Heritage, and Fame, respectively. These were the premier class of fungicide for anthracnose control. The problem is that anthracnose has a mutation at the gene that is effectively inhibited by these QOIs, and it results in a very rapid development of fungicide resistance if we overuse those products. So we have to be very careful about how we use the QOIs in the strategy to control anthracnose. And then we also have a whole bunch of different combo products. And for anthracnose control, the best ones, or the ones to really pay attention to, are the ones that combo up an SDHI plus a DMI. Pick one SDHI, DMI, QOI with a contact. That's why it's XXX. Or the SDHI, DMI with a QOI. Those are all good examples of fungicides that have shown very effective, very efficacious disease control for anthracnose when disease is active. And then when we think about a longer term kind of strategy, certainly our contact fungicides like fluazanam, which is secure, or chlorothalonil, which is Daconil, and the many others that are out there. And then the phosphite materials, Signature Extra, Aliette, and the other phosphites that are out there, Elude and others, are also effective if we're looking at a preventative kind of strategy. And so what I often recommend is that you start with a phosphite backbone in terms of making sure that those phosphites are going out on a regular basis throughout the season. That provides a number of different benefits, one of which is helping reduce the amount of anthracnose you see. We also get some pretty good Pythium root rot and Pythium foliar blight prevention and control from phosphites. So that regular application is going to pick up more than just anthracnose. It's going to give you Pythium. It's also going to help stimulate the defense mechanisms within the plant. There's some evidence that phosphites also affect the plant's metabolism and physiology and help it fight off diseases, which is where we get some of that plant health type of approach with some of the phosphite applications. And so that's another area that you're going to get some benefit from that phosphite backbone. So we want to rely on the phosphites initially as our main preventative, and then we want to use our contacts to help protect plants from the surface. And then we want to look at the SDHIs, DMIs, combos. And then finally, if we've been able to avoid using QOIs thus far, the QOIs are kind of the last ditch. We really don't want to overuse QOIs when it comes to annual bluegrass and anthracnose management, because it's so easy to get fungicide resistance to those QOIs. So we only want to use those at the high rate, short interval, when we have active disease, to really kind of put an extra punch into our fungicide program. And then what I would tell you is that, you know, this landscape is constantly changing year in and year out. We're adding new products, adding new combination products. And so you want to check a reputable source for some efficacy data. And the one that I often go to is the Turf Files website at North Carolina State, because Jim Kearns keeps that updated really well with some efficacy data. And since it's in the transition zone for me, if you want to use the document that's produced by Paul Vincelli and Paul Koch and others out of the University of Kentucky, I know Ohio State produces one. Those are all options for you but you definitely want to look at something that gives you some efficacy data, particularly when it comes to anthracnose, because we want to make sure that, you know, whenever we're in a period of time, and for anthracnose, that window is pretty long, we want to make sure that whatever we're spraying for, that there is some activity on anthracnose, because that's going to help us keep anthracnose at bay throughout the season. And one of the other things when it comes to fungicide resistance, and I learned this just about a month ago, and I thought this was fantastic, and I have to give Kyle Miller from BASF all the credit in the world for this, is you know, we have those FRAC codes that are on the bottles that give you the number of what fungicide class it's in. And they're often hard to remember, but it's a way, if you keep track of those FRAC codes, to kind of keep track of how many applications you're making of products that are in that particular class, because, you know, remembering all the brand names and all the active ingredients gets pretty tedious and frustrating, and it's hard to keep track. But if you keep track of those FRAC codes, you can kind of see that, okay, I'm applying a lot of number 3s, I got to move on to something else, or I need to rotate in other modes of action. It's really important from that perspective. I got a text message a year ago from somebody. I asked them what they had been spraying, and for the past 90 days, they had made an application that every application had a QOI, but all the products were different. And so, you know, it's one of those traps that you can fall into pretty easily. So the little mnemonic that Kyle came up with was Earnhardt and 7-Eleven. So you got Dale Earnhardt, who's number 3 if you're a NASCAR fan, and then you've got 7-Eleven convenience stores, and those are the 3 modes of action in turf that we really need to pay attention to in terms of fungicide resistance. 3 being the DMIs, 7 being the SDHIs, and 11 being the QOIs. And so if we can just remember Earnhardt, 7-Eleven, we've got 3, 7, and 11. It's easy to remember. It's made a difference for me, and hopefully it'll make a difference for you. This is from Dr. Kearns's lab looking at disease development. And I just want to point out that this is the key kind of take home. This is when they receive samples of anthracnose. It's been consistent year in and year out. This happens to be 2017. If you look at 2018 or 2019, they've been getting less bentgrass in North Carolina, but as they get samples in from other places, you see that this window of anthracnose is pretty consistent, and it's pretty much year long throughout the growing season. You get some in the spring right after seed head flush, some in the fall as the plant's starting to grow, you know, slow down its growth, and then in the middle of the summer as the plant is struggling to grow as well. So pretty much anytime the plant is struggling to grow is when you can see anthracnose, and you can see it really all year round if you're looking for it. And so that's why it's so important when we're thinking about our fungicide program that, you know, regardless of what disease we're going after, we want to make sure that the material that we're putting out has some anthracnose control. And then these are certainly trying times, trying to keep everybody healthy and that kind of stuff. And I'm always impressed with the job all of you do in maintaining the surfaces and doing it so selflessly, and getting out there and trying to do what's right for the folks that use your facilities. And so I just want to remind you that you know, we're not the only group that you just can't please all the, you know, everyone all the time. I like to ski, and this was an ad campaign that Snowbird did a couple years back called Disappointment, where they pulled real 1-star Yelp reviews off of Yelp. This is Elizabeth from Dallas, Texas. Are the people who operate the grooming equipment on strike or something? Was hoping for a little more corduroy to dig my skis into. 1 star. And so, you know, you could probably do the same kind of thing with golf. You know, no matter what you do, no matter how well you do it, there's always going to be somebody that's disappointed, and I would encourage you not to let that person get you down. So with that, I'd like to say thank you for joining us today, and happy to take any other questions that have popped up. Thank you so much.
Thank you, Brandon, and a great reminder that, you know, some people are unreasonable. You can't please them. Let's not
That's it.
focus on them. We want to thank PBI-Gordon today. This webinar has been approved for education points. You're going to go to the GCSA website, and you're going to use the code 999-23210-30744 to input for those points. For anyone who's listening to the recording in the future, input the date that you're listening and not today's date. We have a number of other questions here. Let me start at the top of where we did not answer before. What product do you prefer or use to increase your K amounts?
Say that again. What?
What product do you prefer or use to increase your K amounts?
I wouldn't, I don't have a preferred product necessarily. What I would say is that if you're trying to increase K, you want to do it in relation to your nitrogen fertility. Those are often related. And then I've not done a lot of research with MLSN, but I've done a lot of reading on MLSN. That's certainly one strategy. You want to just make sure that potassium is high enough that you're not in a deficient area, and you want to keep that potassium level above the deficiency. And Michael Woods and a number of other people have written about like what strategy you might approach it with, but you just want to use a product that has the potassium along with the nitrogen that balances it out and gets you to where you want to go in terms of relieving that deficiency as quickly as possible, so that you don't have a plant that's sitting there deficient in potassium, because that has been some of the evidence is the potential for increased disease development as a result of that potassium deficiency.
Thank you. When you are combining DMIs, SDHIs, QIs, what rate should you use? First of all, you should always use the rate on the packaging. The label is the law, right, Brandon?
That's exactly what I was going to say. You couldn't have answered it any better. I always do that when I'm in person. I always go, the label is the, and then everybody says law. And that's exactly it. So you definitely want to use what's on the label for the various materials. And that's part of the reason why I don't talk about rates so much as materials, is that I want you to read the label. I think that gets missed way too often when you get used to making applications. You kind of get things in your head. And with the number of tools that we have in our toolbox, there are materials in the same mode of action that have vastly different rates. And so you want to make sure that somebody in your spray group, whoever's making the spray applications, is double checking rates of application prior to every application. So you definitely want to do that. And then in terms of kind of overall recommendations, what I would say is that generally speaking, if we're making a preventative application, then we can be down towards the middle or lower end of a rate range for that disease. So for anthracnose, it's going to be listed on the label of the materials that you want to apply, and you can choose a rate that's if you're going to go preventatively on a regular basis, you can choose a rate that's a little bit lower. If you have active disease, or you're in a period of time that you have to make sure that you get as good a disease control as you possibly can get, then that's where you might select the upper range of the rate range. And if you have active disease, then you're going to want to make sure you're using the shortest interval that you have on the label to allow you to apply as frequently as possible to try to get disease under control. And generally speaking, for most diseases, including anthracnose, if you have active disease and you correctly identify it and you make an application, you should see some improvement in that turf. I'm not saying it's going to get back to good, but you should see some distinct improvement within 3 to 5 days. And if you don't see improvement in that 3 to 5 days, that's where you want to make sure that you've got a sample off to somebody and you're taking a look at and getting an accurate diagnosis. And ideally, you want to get that accurate diagnosis before you make any applications if you can, because that's really an important piece. But with something like this, where you can pull plants out and you can see, you guys can accurately diagnose anthracnose once you know what you're looking for. And then those applications, if you have active disease, those applications need to be at the higher end of the rate range and usually on the shortest interval that you can apply at, which is usually 14 days, sometimes a 7-day interval.
Okay, Justin Wheeler's hand is up. I'm going to unmute Justin. Justin, did you have a question? Okay, I guess not.
Okay.
What role does plant available silicon have in the use of soil nutrient amendment sources of silicon?
So silicon has been debated back and forth in the literature for decades as to what impact it has on the plant and what it does in terms of disease control. That evidence is pretty lacking in terms of decreasing disease. And there's some evidence that it increases the ability of the plant to withstand some traffic stress and things like that. It's not an essential nutrient. It would be kind of in that next set of nutrients that you could probably argue are supplemental nutrients. And so I often kind of like put that into the category of the same kind of thing. Like if you're taking nutritional supplements, you have your vitamins and minerals. If you're deficient in a vitamin or mineral, your doctor tells you, like I was deficient in vitamin D a couple of years back, and they had me start taking a vitamin D supplement, which is fine. But if you can't identify a deficiency, then anything else you're taking is really just a supplement, and it's kind of an anecdotal kind of approach. And in most cases, what I would say in that situation is that you want to just be cautious. You want to be working with reputable companies that have done some product testing, particularly independent product testing. Working with a university or somebody to show you some data of what they were looking for, and then test it yourself. Put out a control plot and see, does it give you a verifiable effect for the money that I'm spending. I think we all need to be good stewards of the money that we're asked to manage, and so you want to make sure that you're doing that. And then if you do see an effect and you do feel like it's improving your turf, and you can define what that improvement is, then I have no issue with those. But I would just say that in terms of repeated, replicated types of research looking at decreases in disease severity, there's a handful of papers that have shown some decreases, and there's a handful of papers that shows not really much effect. And so that's where I really, kind of feel like that's up to the individual user to kind of tease out, well, what are they seeing at their facility under their management conditions? And then that's a situation where I highly encourage you to put out like a car mat, a piece of plywood or whatever on the back of a number of greens, and make sure that you're seeing an effect from something that you're spending money on. And that would go for silicon applications or any other application of any product.
Excellent, thank you. Do you happen to have a slide for bentgrass like your chronology of Bermuda grass putting green disease?
You mean for Bermuda grass?
Yes, sir.
Yeah, so like this, but for Bermuda grass?
Yes, sir.
Okay, so yes, if you go to Turf Files, to Dr. Kearns's disease section, you will find a report from the laboratory, from their diagnostic lab, that every year has a chronology like this that shows you when they were seeing samples in that year. And so you can kind of get a sense as you look at multiple years, and they have them, they get samples from both bentgrass and Bermuda. And so you'll see a list of the kind of the top diseases, and then when they were getting samples throughout the year to kind of give you an idea of when those diseases might be active. Yeah, absolutely.
Excellent. So it seems the biggest take home is manage stress to manage the disease.
Yeah, I would say that the biggest take home for anthracnose disease management is that every management practice that you conduct on a playing surface is either causing stress or minimizing stress. And that balance between those two is going to result in the amount of disease that you tend to experience. And so if you're in a situation when you're struggling, that seems like every year, no matter what you try to do, you get disease, and the fungicides do an okay job, but they never really quite get rid of it. That's where you really need to go in and evaluate the management practices that you're placing on those surfaces and what effect they're having on the potential for disease, and do more to minimize rather than increase the amount of stress. So yes, every management practice that you apply to those surfaces is, potentially a stress to that plant, and you want to do everything you can to try to minimize that to reduce the overall potential for disease.
I think we're all about reducing stress at this particular point in our lives.
Absolutely.
And I'm not seeing any other questions today. Brandon, I want to thank you so much for teaching this for us. Again, thanks to PBI-Gordon, to all of you for participating. If you have questions, please email me, lwick@gcsa.org. Any other closing remarks today, Brandon?
No, if you have questions for me, you can also, in addition to the Twitter handle, you can also reach me at bhorvath, HORVATH @utk.edu, which is another way you can reach out to me and ask questions if you have them. So thank you all for attending. I noticed that we had a really good number of attendees there at the peak of the webinar, and really appreciate you guys all sticking around and asking questions, and hopefully you got something out of this presentation today.
Absolutely. There is a survey as you exit if you provide your feedback. We appreciate that, and we'll see you all online again soon. Have a great day, everybody. Be well.